We're now in the second part of the fermentative gram-negative rods that are non-Enterobacteriaceae family members. And our first part of this lecture, we talked about the Vibrio cholerae organisms, and now we are going to discuss the non-cholera Vibrio species. So these are the halophilic Vibrios, the salt-loving Vibrio. So remember, Vibrio cholerae doesn't like salt, but all of the other Vibrio species do. Vibrio parahaemolyticus causes a diarrheal illness that is less severe than the cholera caused by Vibrio cholerae. It occurs approximately 24 to 48 hours after ingestion of contaminated food, especially seafood. Vibrio vulnificus causes septicemia and wound infections. Predisposing risk factors for Vibrio vulnificus infection include liver dysfunction, such as cirrhosis of the liver, as well as conditions that produce high serum levels of iron, like hemochromatosis. Vibrio vulnificus is a common organism in marine waters, and it can cause eye infections, ear, wound infections, and these infections are usually in association with contaminated seawater. So Vibrio parahaemolyticus is the second most common Vibrio species that causes gastroenteritis. It's much more common in coastal areas. So Vibrio infections, I used to live in New Orleans where it's coastal. There's a lot of oysters, lots of raw oysters, and so there were a lot more Vibrio cases there than there are in other parts of the United States. There's a pandemic strain, O3K6, which has caused global foodborne outbreaks. Vibrio parahaemolyticus is found in aquatic environments. It is halophilic, loves salt, and it requires 1 to 8% of sodium chloride to grow well. It's associated also with marine animal species. So you find it in oysters, clams, crabs, lobsters, scallops, sardines, and shrimp. So all the nice seafood in certain areas of the world and some raw seafood, and you can possibly have a Vibrio parahaemolyticus issue. Most of the gastroenteritis cases are traced back to a recent consumption of either raw or seafood that maybe has been cooked but wasn't cooked well enough. Clinical manifestations are usually a self-limited gastrointestinal disease. So usually an individual will have some episodes of intestinal cramping, vomiting, watery diarrhea with very little to no fever, and it usually goes away on its own. So symptoms normally begin 24 to 48 hours after ingesting contaminated seafood, and you can occasionally find Vibrio parahaemolyticus causing other types of infections outside the GI tract, such as wound infections, ear and eye infections. Usually this is because an individual will have been recently exposed to contaminated water. So if someone was in water and maybe cut themselves in the water, stepped on a shell or something that was contaminated, then it can get into the system that way. Here is an outbreak of an oyster-related illness in Washington State, and this was back in July of 2006. So if you want to go in and eat about this infection caused by eating raw oysters, and it was associated with Vibrio parahaemolyticus. So we do see quite a bit of Vibrio parahaemolyticus infections in the United States. The virulence of Vibrio parahaemolyticus is not as completely understood as it is for Vibrio cholerae. So there's something called the Kanagawa phenomenon, where the organism produces a heat-stable hemolysin that is able to lyse human red blood cells in a special high-salt mannitol media called Wagatsuma agar. So in this situation, it's considered Kanagawa toxin positive. However, there's not that much known about the virulence. There's been a recent emergence of urease positive Vibrio parahaemolyticus strains along the Pacific coast of North America. And here you can see Vibrio parahaemolyticus. I'm going to talk to you about the types of media that you would use to isolate your Vibrio organisms very soon. Okay, here's another one of these little blog spot descriptions on Vibrio parahaemolyticus. Hey there, call me Vibrio parahaemolyticus, part of the Vibrio genus. I'm a gram-negative, oxidase-positive fermenting bacteria. I live in salt water, and you can get me by eating contaminated seafood. I cause gastroenteritis with watery diarrhea, cramps, and fever, and most of the time my infection is self-limiting. So pretty straightforward. Our next species is Vibrio Vulnificus, which is the second most serious species of Vibrio. So the most serious is going to be Vibrio cholerae. The Vibrio cholerae is also going to be the cause of, your worst diarrhea. So this is the second most serious species of Vibrio, found in marine environments on the Atlantic, Gulf and Pacific coasts of North America, and it used to be called the lactose positive Vibrio. So hint, hint, Vibrio vulnificus, lactose positive. It causes two different types of infection. It can cause a primary septicemia, which usually occurs through the GI gastrointestinal route after consuming contaminated shellfish. So again, Vibrio parahaemolyticus and Vibrio vulnificus can both cause a gastrointestinal infection from eating raw shellfish. Now, Vibrio vulnificus has a high association of infection in patients that have some type of liver dysfunction. So cirrhosis of the liver, liver carcinomas, and can have a mortality rate of 40 to 60% because it more commonly infects people that have a predisposing condition. Alcoholics are highly susceptible to Vibrio vulnificus infection because they usually do not have properly functioning livers. Vibrio vulnificus also causes wound infections, again from traumatic aquatic exposure. So wading through water and getting cut in the contaminated water will get into a wound and cause a wound infection, and usually this is a cellulitis. So another one of these little scenarios, hi, I'm Vibrio vulnificus, again, gram-negative, oxidase-positive fermenting bacteria. I'm the most virulent of the non-cholera Vibrios. Like my cousins, I live in the ocean, and I can get into you through contaminated seafood, where I cross from your gut to your bloodstream. Then I can cause septicemia and necrotic ulcers under the skin with up to a 50% mortality rate. Don't worry, that's only if you're immunocompromised or have liver disease. If your immune system is healthy, I can get into your marine wounds, and I can cause severe cellulitis and skin necrosis. Now, the last Vibrio species we're going to discuss is Vibrio alginolyticus. This is the least pathogenic Vibrio species in humans and is not frequently encountered in the clinical laboratory. Again, it's an inhabitant of marine or aquatic environments. It is a strict halophile. It can grow in up to 10% sodium chloride. That's a lot of salt. Most organisms cannot grow in that high of a level of salt. So Vibrio vulnificus is more of a problem in an occupational condition, people that are in constant contact with water, such as sailors and fishermen. So again, Vibrio vulnificus, just like my cousin Vibrio cholerae, I'm a gram-negative oxidase-positive fermenting bacteria. I don't produce cholera toxin though. Some of my cousins and I live in water, especially the ocean. I get into you through cuts while swimming. I can cause skin and tissue infection of your marine wounds. We just love surfers, so if you love the water, come and take me home. Now, laboratory diagnosis. Again, these are facultative anaerobes. They're not fussy at all. Now, glycerol is toxic to Vibrio, so you have to make sure you use a medium that has no glycerol in it. It will grow on MacConkey agar. So remember, with some of our gram-negative rods, one of the characteristic features are that it doesn't grow on MacConkey agar. Organisms like Haemophilus or Pastorella, certain organisms just don't grow on MacConkey agar. All of our Enterobacteriaceae organisms do, and the Vibrios also will grow on MacConkey agar. Now, most of the Vibrio species do not ferment lactose. However, Vibrio vulnificus does ferment lactose. So whenever there's a distinguishing feature among one of the species of a specific genus, that's usually something you need to know. It helps you identify one organism over another. So the fact that Vibrio vulnificus is a lactose fermenter and the other Vibrio species are not is incredibly important. Hint, hint, hint. Vibrio cholerae is usually a comma-shaped organism. The Vibrios are usually considered to be curved. Optimal temperature is body temperature at 35 to 37 degrees Celsius, and the colonies on agars are very similar to the Enterobacteriaceae family members. Very difficult to differentiate the gram-negative rods based on microscopic and macroscopic morphology. You really need those other tests. What sugars does it ferment? Do they produce hydrogen sulfide? Do they produce gas? All of those biochemical reactions are critical for the gram-negative rods. Now, there is a specific selective and differential media that's commonly used for Vibrios, and it's called TCBS agar. And the TCBS is thiosulfate citrate bile salt sucrose agar. Now, this is going to identify your sucrose fermenters. So we already said Vibrio vulnificus is a lactose fermenter, but the other Vibrio species are not. Well, there are some Vibrio species that ferment sucrose. So Vibrio cholerae and Vibrio vulnificus both ferment sucrose. On this agar media is normally a green color. For organisms that ferment sucrose, it drops the pH in this media, and the pH indicator will turn the agar yellow. So Vibrio cholerae and Vibrio vulnificus on TCBS agar will be yellow because they ferment sucrose. Other Vibrio species will just look green, like the color of the media, because they don't ferment sucrose. The TCBS media can be somewhat inhibitory to pathogenic species, so you may need to use an enrichment procedure to enhance your isolation of Vibrios. So whenever you have an intestinal specimen, a fecal specimen, sometimes you have so much normal flora there, you're going to overgrow your pathogen if your pathogen isn't in really high numbers. So remember, we have those enrichment medias where you take your specimen, put it in the enrichment broth, incubate it for 6 hours, and then drop some of that broth onto your selective media to give you a better chance of isolating your pathogen. Now, your Vibrio cholerae and non-cholera Vibrios, you can see Vibrio cholerae on TCBS agar turns the agar yellow and the colonies are bright yellow. So whenever you are trying to isolate an organism in the clinical lab, you are going to use a non-inhibitory media such as your 5% sheep blood agar, usually for intestinal specimens, or a sheep blood agar and a chocolate agar. And you're going to use some sort of selective media, especially with your intestinal patient specimens, because there's so much normal flora in there. You need to select out your pathogens. So TCBS agar is a good selective and differential media specifically for Vibrio. You can use an enrichment media that's a peptone broth that contains 1% sodium chloride, which will encourage some of your Vibrio species to grow. Again, here's two different Vibrios. Your Vibrio cholerae that ferments sucrose that's bright yellow, and then you have your a Vibrio Vulnificus that is a non-sucrose fermenter that is just going to be a green color, the same color as the media. Again, Vibrio cholerae pure culture on TCBS. Now, microscopically, very similar to your typical gram-negative rods. You can do fluorescent staining, like acridine orange staining in a patient specimen. Now, in a patient specimen, the organism, you can see that curve or comma shape to it. Now, Vibrio is going to be easily confused with other gram-negative rods. So all of these organisms can cause intestinal disease. They're gram-negative rods. So you really need to be able to distinguish one gram-negative rod from the other. So again, the Vibrios are usually susceptible to that Vibriostat disc. However, Aeromonas, which is another fermenting gram-negative rod that we're going to discuss next, is resistant to the Vibriostat disc. So it's a good way to differentiate Vibrio from Aeromonas. The inability to ferment inositol separates the Vibrios from the Plesiomonas organisms, which are another group of organisms that are fermenting gram-negative rods. Now, the oxidase positive reaction separates the Vibrios from the Enterobacteriaceae family members, and the fermentation reaction will separate some of the species from one another and will allow you to differentiate your fermentative gram-negative rods like your Enterobacteriaceae members, your Vibrio, Aeromonas, and Plesiomonas, from your non-fermentative gram-negative rods, which we are going to discuss next week. Here's a little chart. Whenever there's a chart, flow chart, some sort of table, it is a good idea for you to know it. So growth in nutrient broth without any salt. Of course, Vibrio cholerae is going to grow, but the parahaemolyticus and vulnificus and alginolyticus won't. They absolutely require salt. All of the Vibrio species will grow in 3% salt. But when you increase the salt, 6%, parahaemolyticus, vulnificus, and alginolyticus will grow, but cholera will not. Cholera doesn't like high salt. And 10% salt, really high salt, only alginolyticus is going to grow, none of the others. Those are key distinguishing features among the Vibrio species. Another distinguishing feature, sucrose. Vibrio cholerae ferments sucrose, Vibrio alginolyticus ferments sucrose, parahaemolyticus and vulnificus do not. Lactose fermentation. Vibrio vulnificus ferments lactose, meaning it will be pink on McConkey agar. And Vibrio cholerae and Vibrio Vulnificus are Voges-Proskauer positive. I highly suggest you memorize this table. However you need to memorize it, my suggestion is to read it over and over, write it down while you're looking at the results, and then if you can write it down 3 times without looking at your paper, it usually means you have it memorized. If you can then write it down quickly each day without looking at your notes, you know it. And it's a good thing to know these types of easy tables, charts, and things like that, because most of the questions on the exams are going to be these types of differentiation, diagnostic questions. So we're going to move on to the third part of this lecture.